lecture, Ian from Proveris is about to talk about about spray pattern, which is much more of the same of the spray analysis as well. So yeah, I'm trying to make it the host that you can share. Yes, you can. Robert and John, you can stay here with us, or if you have another things to do or another lectures in other places, please go ahead.
No problem. I can stay on, and thank you again for the question as well. It's nice to get questions and to sort of engage with the rest of the audience. I think it's always very useful.
Thank you very much.
Welcome. Ian, I think we are in presenter mode. Can you please make the present? Use the 3 dots over down there. Let's try again. Ian, can you hear me? Yeah.
Oh, sorry, I was on mute. I thought you had unmuted me.
Okay.
Can you hear me now?
There we go. So.
Okay, great. All right, so I'm going to share the presentation and go into presenter view.
Let's try slideshow. Yeah. Good. Perfect.
Okay. If I'm in presenter view, can you still just see the slide?
Yes, but you can use in the left side of the screen. There is 3 dots, just right from the right. Yep, and hide presenter view. Thank you. There you go.
Okay. So if I'm in presenter view, can you not? Because I need to see the notes.
Oh, no matter.
Does it project? Okay. Does it project only the slide, or does it show my notes?
It shows both of the slides, the one you're talking about, and then the next. Just what you see, we are see again.
Oh, okay.
Yeah.
All right, I'll try and. All right, so my name is Ian Flaherty.
Let's go.
Cool. So my name is Ian Flaherty. I'm a senior product manager at Proveris Scientific. I'm going to talk to you today about spray pattern and automated actuation for accelerating inhalation and intranasal drug development. Okay, so first of all, I'll talk a little bit about who Proveris is. So we have been around for a little over 25 years, basically developing analytical instruments as well as providing contract test services to people in the OINDP space. So that stands for orally inhaled and nasal drug products. So our work started off primarily with our founder, Dino Farina, developing a tool for non-impaction laser imaging of spray and aerosol products. And then from that came what we now call the SprayVIEW measurement system. So the SprayVIEW has become kind of the gold standard in the industry for allowing researchers to measure spray pattern, geometry, and some other measurements as well.
Jan, I cannot hear you. Please, can you speak loudly?
Okay, sorry.
Okay, there we go.
So, so we are have a global presence at this point. We cover. We have customers basically that are major pharmaceutical companies, contract research, as well as contract manufacturing. Then we work with different device manufacturers, as well as regulatory agencies across the world, including the FDA, the EMA, China FDA, just to name a few. So we are very primarily scientifically focused. So we work on a lot of new applications, have an applications lab that is capable of supporting customers' custom projects. Things like that. So today we're going to talk about, review some of the challenges with OINDP product development, particularly with developing generic products. We'll introduce automated actuation, which is the foundation that our instruments are built on. Talk about how the SprayVIEW system works, and touch on the basics of spray pattern and plume geometry. And then we'll introduce everyone, if you're not already familiar, to the quality by design approach. I'm going to talk about how Proveris fits into that with a little case study. Device design and viscosity of formulation and how that impacts device performance. So, OINDP products are extremely complex to develop, mainly because you have to worry about three main areas: the formulation, the device itself, and then how the patient uses it. So basically all of those factors come together to ultimately what would become a successful combination drug product. So we help our customers primarily in the patient and device realm, and then also helping them with screening different formulations to kind of put all the pieces of the puzzle together. So like I said, for combination drug products, these are typically the challenges in terms of the approval process that we'll see customers run into. So the big one, especially these days, is disagreement with in vitro bioequivalence. So if you look at particularly the market for generic inhaler-based drugs, this has been a huge issue. A lot of projects have gotten killed at the point where they basically just can't get past the approval stage because they can't demonstrate bioequivalence. So this has started to take a turn in a good direction in the past couple of years. We've seen several major generic drugs approved, both for the US as well as European markets. And some other things that we see are just general formulation and device incompatibility. So a lot of times people will have a formulation kind of already set in stone and then move to device selection and find out that their particular formulation does not perform well with the device that they've chosen. And another big one is just general lack of understanding of critical quality attributes. So we'll go over some of those as they pertain to nasal sprays and pMDI drugs, and how our instrumentation can help elucidate some of that for the user. So Proveris is definitely, we see ourselves as a partner through the whole product development cycle. Touch on in a minute, we have our contract laboratory called Proveris Laboratories. And then we offer instrumentation that will pertain to pretty much every step of the development process. So our SprayVIEW measurement system is highly sensitive and can be used as a screening tool very early on in development. But then it also can carry with you through production of your product. It is used primarily with our customers for batch release testing. So to do spray pattern validation, basically to make sure that each batch is performing within spec. And then some of our other systems that are highlighted here are our standalone actuators, our Indizo system, which is for nasal spray testing in kind of a high throughput scenario, and the Canero, which is one of our systems in our pMDI suite of products. So that is for high throughput wasting of pMDI products for through life testing. So Proveris Laboratories, we look to partner with with companies definitely at the early stage, and these are some of the common services that we offer. Usually we start off with a human actuation study where we can understand what the relevant device parameters are for the customer's chosen device, or if they have multiple devices that they want us to screen, and step through reference product characterization, generate data. So I'll go into talking about automated actuation in spray pattern and Plume, and just give everybody a quick fundamentals overview if you're not already familiar. So our Vereo automated actuators are used in a whole variety of different tests in our contract lab as well as at our different customer sites. So we have currently 4 different models that will allow you to test different device types. So pMDIs, vertically actuated nasal sprays, oral sprays, side actuated nasal sprays, dual squeeze nasal sprays, just to name some of the major ones. So these are common tests that our actuators would be applied to. Vitro tests. So when you talk about nasal sprays, some of the key parameters as referenced in literature that you want to be able to control are stroke length, especially for a vertically actuated nasal spray that often ends up being the most important parameters. So with our actuator, you have the ability basically to repeatedly and reliably actuate the device to a set stroke length. So once you kind of find out what the optimal value is for your device, then you can kind of continue all of your testing using that same profile. So you'll see here just an example of how metered shot weight varies with stroke length. So you'll see once you get up into that range of around 5 millimeters, your targeted shot weight for the device. So you can see varying the velocity, or sorry, varying the stroke length has a big impact on how your device performs. So velocity is another important parameter. In software, you can control that as well. So velocity has the potential to greatly impact spray pattern and plume geometry measurements, as well as droplet size distribution. So these 3 videos are just examples of extremes, so low, medium, and high velocity. And you can see how different spray profile is. So this orientation would be set up for a plume geometry measurement. It's actually just a live video from a particular measurement for each of these 3 devices. When you go to the side of pMDIs, so like inhaler drugs, another factor that's very important to consider is shaking parameters, especially if you're working with a suspension formulation. Sometimes it can settle very rapidly within the seconds in some cases. So making sure that you have a good shaking methodology, as well as minimizing the shake to fire delay. So once you complete shaking, firing that device immediately is important. And this is an example of some data that we generated showing how the dose content can greatly change based on proper shaking. So that trend line in blue basically shows that if you don't perform any shaking, all of the API is already settled at the bottom of the canister. So your first shots through life are going to dispense a huge amount of drug. And then after that, basically, you've essentially used most of the API up. So the rest of your shots are going to be much lower than intended dose. as you can see, shot weight doesn't change because you're still getting excipients and other things expelled with each shot. The valve itself is metered, ensuring that have that proper distribution of drug is very dependent on shaking. And then just proper actuation in general for pMDIs can greatly change the performance. So you can see on the left, we have a fully open valve, which has that nice burst, pretty even distribution in terms of the intensity of the image. Now on the right, you have an example of one where the actuation was caused the valve to only partially open. So you get some what we call sputtering. So it's just another example of how using automated actuation is important to product performance and to reducing any variability that can be introduced from manual human actuation. So how do we get these parameters to use on the system in terms of velocity and stroke length and things like that? Generally, what we like to do is start off with what we call a human actuation study. This uses a special instrument that we call the ergo. There's a mock-up of it here with pMDI. But basically, it allows us to have a human actuate the drug and measure parameters like velocity, hold time, stroke length, etc. So we can do a study where we get a certain number of patients to get a good value and basically kind of come up with average values for these parameters that we can then use for automated actuation going forward. You can see here just an example of how variable manual actuation can be. So everything on the left here are profiles from actual humans actuating the device. You can see hold time, velocity, and stroke length varies quite a bit. Whereas on the right, this is just an example profile from one of our actuators. So it's very, very unique. So once those parameters are kind of zeroed in, then all the testing going forward happens with very, very low variability. Just another thing to point out is the force profile itself. It can kind of be a sort of a signature or like a fingerprint for the device to kind of help you visualize different parts of the actuation process. So particularly with some complex devices. So recently there's been definitely a lot of popularity in what are called unit dose devices. And that's an example of one on the left here, basically designed to hold only one dose so that when it's administered, the device is thrown away. And this particular device is made by a company called Aptar, and it's very popular, but you can see the actuation profile is fairly complex because there's a lot happening. This green trend line shows the force. So initially there's a tab that you need to break. So that requires a good force peak. And then once that tab's broken, you have the second region where the needle's hitting the rubber stopper, and then you get to your delivery stroke, which is that gradual rise in force right there. So this just shows that for each actuation record, you get the ability to kind of look at this data and start to see trends and be able to overlay different force profiles and find out of specification shots or things like that. Okay, so now we'll talk about SprayVIEW. So SprayVIEW utilizes our automated actuator technology, but it basically adds in a couple other elements, a semiconductor laser system and a high-speed camera so that you can visualize dynamic aerosolization of the spray throughout the full spray event. allows you to generate time synchronized results and quantify certain measurements like spray pattern and plume geometry, and associated secondary measurements that come with those. So it's a very robust technology, very reproducible. So you'll see basically it's a good tool to kind of throughout the product development process, because you're going to get very consistent data throughout. You'll be able to highlight very subtle differences or subtle changes in device performance. So just break down the components of the system. Basically have a environmental enclosure on top that provides airflow to move everything out of the cabinet after a spray event happens, and it passes over a set of filters. We have our high-speed camera system, Vereo actuator. So depending on the device type that you're using, there might be a different version of the Vereo in there. Right now it's shown with nasal spray with our Vereo NSX actuator. And then we have a laser light sheet system, which is just a semiconductor laser that basically illuminates different planes of the spray and allows the camera to image it. Everything is run from our Vereo software. So basically, and just to kind of break it down, it's a non-impaction imaging system. We have encoders that record the distance from the nasal tip or from the mouthpiece edge, which helps with setting measurements at different distances as required by different regulatory bodies. So with spray pattern, generally they require you measure at 2 different distances away, generally 3 and 6 centimeters. And so our software basically has a built-in algorithm that automatically will calculate the spray pattern of the true shape. So it actually is fairly complex in terms of being able to work with variable patterns or ones that aren't perfectly circular. So it doesn't use like a fitting technology. It actually calculates the true perimeter of spray pattern shape. So these schematics kind of show what the differences are in orientation and how things are set up for spray pattern versus in geometry. So you can see it's basically orienting the laser to create, to highlight different planes of the spray. So in this case, we're highlighting a cross-section of the spray from a vertical nasal spray. You can see the fan of the laser is shaped like that, and then the would image down. Versus over here, we have a pMDI where the laser is basically cutting right through the spray. So you get this cone-shaped cross-section projection, which is what we call a plume geometry measurement. So for a spray pattern measurement, your main data points are basically spray pattern area, so the total area of the pattern itself. The ovality, which is essentially how circular or how oval the pattern is. So obviously, ideally, you want be of 1, which would be a perfect circle. And then you get the max diameter, minimum diameter, which is used to calculate the area. This measurement is generally performed at 2 distances. So on the right side, you'll see the position in red, the force profile in green. Then this graph is the intensity. So this is relative image intensity during the duration of the spray event. So you'll see when the spray starts, the image intensity spikes and then gradually falls back down. The spray concludes. For plume geometry, your parameters basically are you measure the plume width at a certain distance, usually at 60 millimeters or 6 centimeters. So that would be this measurement right here in terms of how wide the plume is at that distance. The plume angle, which you can set these crosshairs in order to calculate. And this is generally performed at a single distance. And you can utilize the video playback, particularly in plume geometry, to get a good idea for how the aerosolization process develops throughout the whole spray event. So it's a good kind of qualitative measurement that you can get as well. And this is an example kind of highlighting that last point in terms of showing two very different devices. So on top, we have a unit dose nasal powder, which is a very quick dispensing of the drug, very narrow angled plume. And so you'll see the corresponding image intensity is a really quick spike, rapidly decays, versus a softness inhaler bottom here, which is a newer type of inhaler device, basically, that creates a very long duration spray and very uniform. So you'll see here, just with the same time scales, the spray duration is well over a second compared to the unit dose nasal powder, which is around 70 milliseconds. So here's another example of how intensity profiles can illustrate spray dynamics. So in terms of in vitro bioequivalence, topic, right here we have two different devices, a generic and a reference, where the plume width and angle are almost identical. If you look at the intensity profiles, you'll see some distinct differences. And you can see that in the actual raw video, the plume geometry, that they're actually not performing the same despite the plume geometry measurement. So it just shows you like the depth of data that you can glean from different aspects of the plume geometry measurement. So you'll notice the key difference here is there's a secondary firing event happen with this generic drug. So that would indicate either clogging or potential issues with the valve design. And one final kind of new application that is being used for SprayVIEW is to measure the velocity of the plume. So this is a new measurement that's been indicated by the FDA as something that would be acceptable for bioequivalence study in terms of submission for a generic drug. So what you're seeing here is measurements of the velocity for a pMDI and a softness inhaler. So we actually use the plume geometry measurement to be able to achieve measuring the velocity as well. So we can actually define the time 0 point as the time point just before the spray event starts. And then we can measure how much distance the plume, the leading edge of the plume, covers throughout the field of view in order to calculate the velocity. So in terms of quality by design, so you may have heard that term before. It is recommended in terms of what the FDA has laid out in their guidances to kind of follow this process, but it's a very kind of vague process. So we've adapted the methodology into what we call Proveris by design. So we use this with our contract service projects, and as well as help guide our customers who have purchased instrumentation from us to kind of follow these guidelines. So it's basically the principle is broken out into these three areas that are called the design space, the control space, and the operating space. So mainly the design space, you're investigating essentially how does the product perform in actual patient usage. So a direct quote from the FDA is that basically critical formulation attributes and process parameters are identified by determining the extent to which any variation can affect the quality of the drug product. So that's basically kind of baselining performance of the drug. So then you move into the control space where you can set up scenarios and run experiments in order to identify how certain critical quality attributes are affected by changing certain parameters. And you can kind of identify which parameters have the most impact on the product performance. And we'll outline an example of that in a case study shortly. So ultimately you arrive at the operating space, which is essentially the statistically best set of parameters that enable you to accommodate any natural variability in the process and the formulations. So for generic products, the operating space generally should be within the control space and allow basically for the reference product to be tested with the same set of actuation parameters. So that's your ultimate goal, is coming up with a method of defined actuation parameters where your test device or your soon-to-be generic drug performs the same as the reference product. And that's why, in general, we approach generic drug development as starting with a thorough reference product characterization. So in here is where we run a whole host of the different in vitro tests, things like shot weight, spray pattern, plume geometry, droplet size distribution, things like that. Full picture for how the reference product, how it operates essentially. Then you move into device selection and then start control screening experiments. So that's when you're comparing your test product to the reference at varying different parameters. And then ultimately you end up with method validation. So we talked about this a little while earlier, but getting parameters to start your in vitro tests with usually starts with a human actuation study. That's where you translate those basically human actuations into parameters that you can enter into our software and have actuation, the Vereo actuator fire the device. So that reference product characterization can be kind of broken down even more to these items that we like to follow. So human actuation study, like we just talked about, and then you move into shot weight. So to us, shot weight should definitely be the first test that you run, just because it's the most indicative of the performance of the device. If it is not consistently delivering the same shot in terms of weight, then you're going to have a problem from the beginning. And what you do with formulation and what you do with other things is not really going to affect the performance very well. And then from there, we like to recommend people move into plume geometry to confirm that the spray is aerosolizing uniformly and properly in terms of comparison to the reference product. And from there, we move generally into spray pattern, and then followed by droplet size distribution. So now, I'd like to talk a little bit about.
Jan?
Yes.
I wish we could have time for case study right now.
Okay, am I over?
Yes, you are. But it was. This is what happening when you're very concentrated in the great deep into the spray data or something. Yeah, you're losing the time.
Yep.
Actually, I want to thank you very much. Could you send us this case study?
Yes, of course.
Okay, it will be better, and I will send this to the attendees that actually waiting to see the case study right now. I'm sorry to interrupt, but this is the end of the day. I want to thank you very much for wake up early for us and for the fabulous lecture. I would like to share some thank you page. Please, if you are not following us over the LinkedIn, please do. We would like to just want to say thank you. That's what I'm trying to do. There we go. So you can find picture from the day over the LinkedIn, and I just want to say thank you very much for you, the client that arrived and was here all day with us, and also to you guys, lecturers from all over the world. We have actually from Europe all over and US, and it was fabulous to be with you here and to host you here in Dr. Golik Academy Pharma Symposium. It was a pleasure. I'm gonna move to Hebrew because this is the audience for me. So thank you guys. תודה רבה, ענקית, גם לצוות של דוקטור גוליק שעזר לי להרים את היום הזה. לסגל המרצים המומחים מהעולם כבר אמרתי, אבל גם לכם שהגעתם לכל ההרצאות, מי מכם שהגיע ליום שלם, מי מכם שבחר הרצאות ספציפיות. ראיתם כאן היום נגיעות מתוך סל פתרונות שיש לנו להציע. אני מזמינה אתכם לשתף אותנו בכל אתגר שיש לכם. בין אם האתגר שלכם הוא עבור פיתוח, או בין אם האתגר שלכם הוא עבור תוצר שהוא שונה מהתכנון שלכם בתהליך הייצור. יש לנו אפשרות לבצע מדידות, יש לנו אפשרות לנתח נתונים, יש לנו אפשרות להציע לכם תמיכה בכל דרך, ויש לנו מחלקה לתמיכה במו"פ. אם אתם מעוניינים לקחת קורסים בגוליק אקדמי, או להכשיר את הצוות שלכם בגוליק אקדמי, יש לנו את היכולת להעביר לכם קורסים ספציפיים, שיכולו לתת להם הכשרות hands-on על המכשירים. תרגישו בנוח לפנות אלינו, אנחנו כאן, דוקטור גוליק, בשבילכם, עם כל הפתרונות, גם לעולם הפארמה. תודה. Thank you very much. ושיהיה אחלה המשך יום לכולכם. ביי ביי.